Okay. Assalamualaikum warahmatullahi wabarakatuh, everyone. I hope you can hear my voice. This is the live stream, so I suppose that you can always, join me if you are available now. If not, then maybe you can view the videos in my YouTube channel, and it will be available up to 11:59 tonight. So please don't forget to click on the attendance link that I have provided. So you can always click on the attendance link that I have already provided to your class representative. So today, this is the, I can say, the replacement class for the immunology that we have missed maybe last week or last 2 weeks, I forgot already. But yeah, in this class, we expect you to learn a few things, especially when we're dealing with the hypersensitivity. Some people say it's allergic reactions, things like, eczema, contact dermatitis. They are all allergic to certain different antigens that is not, cannot be processed by your cells, right? Okay, so I hope you can find or you can call your friends to come and join me for today's replacement class. So we go to the hypersensitivity. I hope you can hear my voice because there's lots of distraction out there. Hypersensitivity is a kind of immune reactions that offer protections against infectious agents. They are metabolites and other harmful substances in a healthy individuals. In other words, it's a protection mechanism to avoid yourself from dying. All right, so your body have a protection barrier or mechanism to avoid these foreign bodies or antigens to invade your immune system. However, in some cases, immune response, instead of providing protection, they can cause deleterious reactions and can cause the extensive damage or even death to the host. So this is important for you to know that failure in maintaining the protection by your immune cells can cause lots of consequences later on. Okay, injuries such as immunological reactions, hypersensitivity, allergic reactions, what else? Anaphylaxis. They are all the types of hypersensitivity or reactions that can occur due to failure to protect your body. So what are the definition? Hypersensitivity reaction is a state of altered reactivity in which body reacts with an exaggerated immune response to what is perceived as a foreign substance or an antigen. Okay, in the book we call it foreign substances as an antigen. And hypersensitivity reaction is found in immunologically sensitized individual after subsequent contact with same antigens, resulting in exaggerated or little process of immunity. In other words, this happening continuously until you are given or your body already have an information to attack or counter the antigens. In other words, the antibody is the key players to bind to this antigen and then immediately remove it from your system. And this can be localized to one area or generalized. It may include rashes, itching, high swelling, difficulty in breathing, and also low blood pressure. And hypersensitivity, aka the allergic reactions. The concept of hypersensitivity was introduced by two French scientists called Paul Portier and Charles Richet They found that localized reactions of bathers in Mediterranean to sting to Portuguese men of war, we call it the jellyfish, was due to the toxin. So this jellyfish was able to cause the allergic reactions in people who are taking a bath inside this Mediterranean Sea. And from that event, they found out, okay, these jellyfish or certain type of organism can produce a toxic substances that is harmful to our body system. And hence, these hypersensitivity developed. And also people used to say that this is a kind of allergens. The initial contact with the antigen leading to synthesized B or T lymphocytes, your defense immune cells, in a sensitized individual is also known as the sensitizing dose or priming dose. All right, so this is the first contact that, you know, when the antigen is quite new, the first time they enter in a form of pollen, for example, in a form of bacteria or viruses, they are all the antigens. So the B and T cells, their functions to recognize these cells, these antigens, and start to produce your proteins and also the antibodies to counter this attack. The second contact with the same specific antigen resulting in hypersensitivity reactions are called as the shocking dose. Okay, so this is the second one. If your body failing, or if your body failed to produce antibody against that particular antigens, so it can cause these shocking, hypersensitivity reaction. We call it the shocking dose. This is only the terminology that we commonly use in the textbook. What are the types of hypersensitivity reactions? If you want to know, on the basis of the time taken to develop the effector molecules during the course of reaction. So we examining since the initial contact with our cells, all right? The first point that our cells recognize this antigen, that is the first time, that we calculate the immune response. Immediate hypersensitivity reactions appear and recede rapidly, induced by antigens or haptens through any routes, antibody-mediated reactions. So these include the reactions like people with penicillin, immunocompromised or non-tolerance to the what we call the penicillin. So they're very sensitive to the penicillin. So it can cause the anaphylaxis. I think you already covered this in your PBL last time. Atopy, hemolytic diseases and autoimmune diseases, others reaction, and also serum sickness is an example of immediate hypersensitivity. It means that once you are exposed to this type of antigens or haptens, they can immediately cause the reaction, and it takes a few seconds to few hours to develop, right? Other than that, we have the delayed type hypersensitivity reactions, and these appear much slower and will take some time to process. This is due to the complexity of the antigens for the lymphocyte to recognize and produce the antibody. Okay, however, it lasts longer. It means if you got the antigens coming to your body for the second time, third time, or whatever time, so still you have the memory B cells to say that, okay, we have we have seen these type of antigens before, so it's quite easy for us to immediately kill them. And this is, for example, induced by the infections, bacterial infections, viral infections, injection of antigens due to some treatments, or haptens intradermally or by skin contact. They are all, causing a delayed type of hypersensitivity, right? Okay. So what is haptens? Haptens is the molecules when we combine with the larger carriers such as the proteins, it can cause the productions of the antibodies that is binding specifically to it, okay, in the free or in the combined states, that is hapten. Similarly, like the antigen, okay, it's just that they require a carrier for them to travel, okay, but the antigen can travel by their own. For example, delayed hypersensitivity includes the contact dermatitis, tuberculin reactions, okay, tuberculin infection by the TB bacteria. Next, the gel and comb classification of hypersensitivity reactions. These are the two scientists that revised the classification. They are both immunologists that try to understand the pathophysiology of the immunological responses that include this hypersensitivity. Some books, they mention 4 types, but based on the gel and combs, they come out with 5 different types of hypersensitivity. Type 1 is the IgE mediated or anaphylactic type of hypersensitivity. Type 2 are known as cytotoxic hypersensitivity. They are cell mediated as well. Okay, type 3 immune complex mediated. Type 4 is the delayed type, and type 5 is the stimulatory. So I will brief it to you one by one. For the type 1, this is the most common that we can see happen in people. They are all related with the histamine production, and this occur when the antigens or the allergens attach with the antibodies, especially the IgE, that is present on the surface receptor of mast cells and then basophils. Subsequent exposure to the same antigen or allergen cross-link to the antibody, the IgE, and start to initiate the release of various active mediators that leading to the complications or certain diseases. And this reaction usually take within 15 to 20 minutes, so it's a immediate type. So it occurs very quickly. People with, that sensitive to penicillin also can have this type of hypersensitivity. So this is an image to illustrate the mast cell that has been activated and how this mast cell functions to trigger the release of a few mediators after the allergens or the antigens start to bind to the surface. The ECF is example. ECF, I can see here, what is ECF? ECF, these, if you have this, if I'm not mistaken, is eosinophil chemotactic factor, and NCF, the neutrophil chemotactic factor. We have PAF, the platelet activating factor. They are all the mediators that regulates the development of type 1 hypersensitivity. If you look at the structure of the mast cell, you can see start from the top one, antigen binding to the Y-shaped antibody, okay? So the Y-shaped antibody is the IgE. All of the antibody, in all books, all textbooks, we have one of the distinguished features is the Y shape. They have the wing and also they have the neck at the bottom. Okay, so this is the characteristic of the antibody. When the antigen bind to any of those one of those wing or hand of the antibody, you can say it start to trigger the signals for the mast cell to produce lots of mediators, okay, only by binding of this antigen to the antibody. Mediators like the histamine is the most commonly reported, proteases, lots of enzymes, chemotactic factors, ECF, NCF, they are all considered as the primary mediators released by the mast cell. However, the other reactions also occurring if the antigen and antibody binding have some signals and affecting the nucleus, they can have a different pathway, and they can also secrete different type of cytokines or mediators, and we call this as the secreted cytokines. All right. Apart from that, we also have the mediators that is produced through the membrane phospholipids. That includes the arachidonic acid, steroid-based molecules. We have also the platelet activating factors for different functions. Leukotrienes, prostaglandins, they are all classified under secondary mediators that functions in the regulations of the blood vessel, all right, the vasodilation or vasoconstrictions. So you can see within a single cells, okay, within a single mast cell, there are lots of activities or lots of functions that they can elicit in order to regulate the immunosensitivity or hypersensitivity. The PAF, the platelet activating factor, that are known also as a lipid mediator because they are made from the lipids, involved in several allergic reactions. It's being released by lots of immune cells, not only by the mast cells. Eosinophils also producing the PAF, neutrophil as well. Okay, so they exert their effect most of them, okay, upon specific binding. Okay, antigen and antibody binding is essential for the activation of these cells. All right, so I just give a link. Maybe you can just type in and find the videos, but yeah, this is for your own or additional information. Okay, so what are the mediators that we are looking at? The most important one, which is the histamine. It occurs in granules of mast cells and basophils. The release of histamine result in vasodilation of the blood vessel. Of course, it increases the capillary permeability and also smooth muscle contraction. Right. So when people are secreting lots of histamine, they are regulating how the blood vessel works. Right now, you are dilating the diameter of the blood vessel. At the same time, the smooth muscle or the endothelial cells surrounding the blood vessel now contracting. Okay, so you have both muscle contraction and also vessel dilation due to the histamine release. Another signaling or mediator, we call it leukotrienes. They are slow reacting substances as compared to the histamine and do not occur in the preformed state, but they produce during the reaction, shows increased vascular permeability and also smooth muscle contraction as well. It's additional signaling mediators that also involved in the hypersensitivity type 1. ECF, this is a long peptide chain that exists in the preformed state. Preformed state means the structure is not complete, means there are partially ready molecules, not like histamine and leukotrienes, where there are full molecules that are already fully synthesized. Okay, preformed state means they need another step to complete their structure, and also it's partially, you can say that they are partially ready. However, they also have the specific functions. It attracts eosinophil and play a prominent role in the mediation of allergic reaction. Prostaglandin and thromboxanes, they are all mediators as well, closely related to the leukotrienes and help in how platelets aggregate, especially during hemostasis. Excuse me. Serotonin is found in mast cells and platelets. It released during anaphylaxis. Increase the vascular permeability, capillary dilation, and smooth muscle contraction are caused by the serotonin. Excuse me. All right, and some other molecules are also important, but I just highlight these 5 mostly reported mediators that is specifically involved in the type 1 hypersensitivity. Then we can understand the inflammation. Okay, the next step is the reactions of the body. Okay, what happens next? Excuse me. So a non-specific exaggerated physiologic response, which involve the vascular response and cellular response by phagocytic cells to infection and injury. So this is also one of the manifestation or clinical manifestation from the immunological response. And mild may be only edema. Reaction is triggered by mast cell basophils. If inflammatory cells are present, many eosinophils. Okay, inflammation there are of two types: acute and also chronic. Acute can occur very quickly, and the chronic, you can see, can last like few days to weeks. Type 1 allergic reaction, it can cause by the skin contact to the pollens from the plants, poisons, toxic substances from maybe mushrooms, latex, animal scratches, animal fur, feathers. Or it can be due to the injection, like the bee sting, ingestion of some antibiotics, penicillin-based antibiotics, the broad-spectrum amoxicillin, for example, nuts, shellfish, almonds, peanuts. They all can cause these allergic reactions. Okay, this is just an example, and you can see the development of hive, difficulty in breathing, and some of these reactions that can occur. Can give you some of the clinical images that is, illustrating the anaphylaxis. You can see like the lips also become bigger or swollen. And this also, one of the signs are the inflammation. You can see, and you already learned this before in the inflammation class. And I don't think I can go into details, but my focus today is to guide you for the immunological responses. Of course, inflammatory response is part of this mechanism. And if you can see that the number one, I'm not sure if you can see here, the damage tissue release histamines, increasing blood flows to the area. Okay, that's the first stage. And then they are sending lots of mediators or triggering some other cells, especially the immune cells, to produce the histamine. Maybe polymorphonuclear cells, okay, PMCs, like the neutrophils that have the granules. Okay, they can release all these histamines to signal saying that, okay, we need to do something. The body are in the dangerous state. So they are producing these mediators to help the body recover from the attack. The presence of phagocytic cells, okay, macrophages is important as a key players to engulf of this bacteria that causes the inflammations. Opsonizations, and in other words, for today's class, is the coating of antibody or complement to facilitate the phagocytosis process. For example, opsonies, we have the C3b, C4b, complement system, C5b, fibronectin, leukotrienes, immunoglobulins, or AKA antibody. And then the next process is the engulfment. All right, this is achieved through the amoeboid motion, and then making the phagosome. After that, they degranulate and digest this molecule. So opsonization is like the coating of our antibody. Okay, one way or the first activity in order to recognize the antigen. In other words, the opsonization is an immune process that uses opsonin to tag the protein or foreign pathogens for elimination by the phagocytes. Okay, they are helping the macrophages to kill the invading bacteria. And without this opsonin, such as the antibody, the negatively charged cell wall of the pathogen and phagocyte will repel each other. Okay, so this is one of the mechanisms. I cannot show you the animation, how, okay, unfortunately, but maybe you can just understand it easily from the figures. What are the diseases related with the type 1 hypersensitivity? One of those are urticaria and angioneuritic edema, asthmatic attack, also type 1 hypersensitivity, hay fever, insect allergy, serious or certain toxic materials, maybe snake bite or bee stings, they are, can cause an allergy. Edema of the larynx with airway obstruction may also occur. Hive, what are the hives? Urticaria, okay, these are the red itchy welts, okay, that result from the skin reactions. And also this ring or the rashes can vary in size and can appear and also fade repeatedly as the reaction runs its course. The condition is considered chronic hive if the redness, okay, appear more than 6 weeks, okay, and can recur frequently over months or even years. They can come and go anytime. Allergic rhinitis, okay, some people saying that this is, aka hay fever, okay, this is the allergic rhinitis. Some book they call it hay fever, they are all the same thing. They are the allergic reaction that causes the sneezing continuously, congestion, itchy nose, and sore throats. Things like dust mites, pollens, pet dander, mold, insects, they can cause this hay fever symptoms. So be careful, all right? Type 2 hypersensitivity, there are cytotoxic reaction or cytolytic. One of the mechanisms is that they are manipulating the complement-dependent reaction. In other words, the body needs to produce a certain complement or attachment molecules so that the antibody specific to that antigen can be produced. And mechanism, things like transfusion reaction, erythroblastosis fetal, autoimmune hemolytic anemia, certain drugs reactions can induce this hypersensitivity type 2. Hemolytic anemia, what are they? If you have learned or have you come across this disease, it's a type of disorder in which the red blood cells are destroyed faster than they can be made. Means the rate of destruction is, quicker than the time taken to produce them. All right. So the destruction of the RBC called the hemolysis. You know that the red blood cell carry oxygen to all parts of our body, and if you have lower than normal amount of RBC, you will have this anemia. All right. So this is type of autoimmune disease. So it's, commonly cause fatality, right? Even though here the fetalis one, erythroblastosis fetalis is a type of hemolytic anemia as well, but it occurs in the neonates or in the fetus, right? And it caused by the transplacental transmission of the maternal antibody to the fetal RBC. Okay, the transfer of this maternal antibody to the baby. So it can cause some reaction, because not all the mom's antibody is suitable or stable when it comes into the baby. Some of them can cause reactions, right? So this is a type 2 antibodies, type 2 hypersensitivity. Another type inside the type 2 as well, we have the antibody-dependent cell-mediated cytotoxicity, in short ADCC, and this commonly occur when you want to have the donor, when you have the transplantation, where you seem to have the graft rejections. GVHD, this is the most graft versus host disease. Okay, this destruction of targets too large to be phagocytous, such as parasite or tumor cells, too much to handle. So yeah, it's compromising the capability of your immune cells to kill them. That's a cell-mediated under type 2. And also under type 3, we have these antibody-mediated cellular dysfunctions. Diseases like myasthenia gravis, where you lose your capability to contract the muscle, lead to loss of muscle or muscle, what do we call it? Muscle cells. Okay, your activity of muscle is reduced. Graves' disease, hyperthyroidism, overproduction of the thyroid hormones, T3, 3-iodocyanine can cause the bulging of your eyes, right? So this is the type 2 as well, but under the antibody-mediated cellular dysfunctions. This is example of the complement system. I thought I already gave this lecture in the pathology class before, but I'm not sure if you already have this explained with me. Anyway, I'll just briefly discuss. The complement is another way that our body can produce antibody, right? Because not all the antigen is possible for our body to recognize. So we need a complement system. In other words, we need a help from another type of protein to help the antibody to work and produce, and kill the antigens. In this case, it's the in red. Okay, I think the red one is the antigens or the target cells. So this is the schematic illustration of 3 different mechanisms of antibody-mediated injury in type 2 hypersensitivity. A is the complement-dependent reaction that leads to lysis of cells or render them susceptible to phagocytosis. In other words, complement system is the manipulation of the immune cells to kill the target cells, right? So as I mentioned earlier, not all antigens are capable to be recognized by the immune cell. It will take some time, all right, to learn the structure, to learn what type of protein, so that it will be better to be killed, or it can be easily killed by the phagocytes later on. All right, we have lots of complement system. We have C5, we have C3b. Okay, so they are like the C5 to C9. Okay, the first one, you see that the target cells plus the antibody. Okay, the Y binds to the antigen on the target cells plus the complement system. As I mentioned earlier, you still need the complement system, another protein or molecules that is essential. Without this complement system, if you just rely on the antibody, that is impossible for the body to get rid of these target cells or invading pathogens. So now you have the complement system, like complement C5 to C9. They will form a membrane attack complex and cause the lysis of the target cell. That is one way, right? Another way, we have C1423. They are manipulating a different way of producing a small C3b receptor that is bound to the antibody, and later on this complex can be recognized by the phagocytes. In other words, you are helping the phagocytes or macrophages to recognize the target cells, because the target cells are very clever. They can cheat. Okay, they can cheat our immune cells so that they won't be recognized. Right? If they are successfully, avoid these things from happening, you are in danger. Right? So our body has been programmed to know whatever viruses, bacteria, they are invading our system. There are a special system, okay, to learn and to get rid of these pathogens. Okay, one of those clever systems is the complement system, as what we are mentioning now. Other than that, we have the ADCC, the antibody-dependent cell-mediated cytotoxicity, ADCC. IgG-coated target cells are killed by cells that bear Fc receptor for IgG. For example, natural killer cells, NK cells, and also macrophages. This is another clever system for our immune cells. For example, here we focus on the NK cells, natural killer cells, how the NK cells can detect the target cells and kill themselves immediately. All right. So of course, now they are utilizing a different portion of the antibody. As I mentioned earlier, antibody is the Y shape. You can see the Y structure. So they have their hand waving at you. So these are called the AB receptor. Okay, the two, like the Y, okay, like this one, like peace sign. Okay, they are A and AB receptors. Some antigens use this for the binding. And then you can see like the neck, okay, or the straight line. Okay, Y, okay, make the letter V, but then the branching downwards, okay, the straight line from the V that makes the Y. So that stick, okay, that straight lines is another receptor, another portion of the antibody. We call it as the Fc receptor. Okay, so the Fc receptor only allow a certain antigen to bind to it. Okay, they don't allow all pathogens to bind to it. Okay, certain molecules can bind to only AB receptor of the antibody. So in this case, we are utilizing the Fc receptor of the antibody so that the NK can bring or can bind this antibody, and then later the AB receptor can bind to the target cell and immediately kill them. So by this way also, this is another clever way that the body adapting to recognize and kill the invading pathogens. So there, lots of other mechanism that is happening in our body that we still did not know. Okay, this is only part of them. And this is using the anti-receptor antibodies, using the antibody to acetylcholine receptors. This is the NMJ, the neuromuscular junctions at the tip of the synapse, or what we call it, your nerve cells. Okay, you have these acetylcholines at the neurotransmitters. They are providing the signaling molecules to be passed on to the next target cells. But in this case, you can see the anti-receptor antibodies disturb the normal functions of the receptors. And in this example, we have the acetylcholine receptor antibodies that damages the neuromuscular transmission in myasthenia gravis. So the failure of these antibodies to be recognized, or the damage of these acetylcholine receptor antibodies, they causing the myasthenia gravis, loss of a capability for the muscle to contract, because now the impulse is inhibited. That one is a type of what we call the anti-receptor antibody. The third one is the type 3 hypersensitivity. It's involved the immune complex mediated. The reaction types include the Arthur's reaction. What is Arthur's reaction? The Arthur's reaction is the result of localized inflammation of the small vessels, like the vasculitis near the injection or the bite site. Okay. Serum sickness is the reaction more. It's like the allergies. Okay, the immune system reacts to the drugs that you are taking. And also these drugs are containing the proteins used to treat the immune condition. It also reacts to the anti-serum. Okay, the anti-serum, which is the liquid part of the blood that contain antibodies given to a person to help protect them against germs or poisonous substances. Collagen diseases, the term previously used to describe the systematic autoimmune diseases like rheumatoid arthritis, RA, or systemic lupus erythematosus, and systemic sclerosis. They are all the autoimmunes that attack the collagen production. All right, so these are all the type 3 hypersensitivity. What are the toxic complex diseases related with the type 3? They include acute glomerulonephritis, SLE, necrotizing angioitis, RA, progressive systemic sclerosis, dermatomyositis, among others. And this is how the systemic type 3 is happening, involving the immune complex hypersensitivity. You look at the phase 1 where the immune complex formation from the B cells binding to the antigen, and after that they got triggered, the plasma cells will produce the antibody, and then this antibody will combine with the antigen again to make a antigen antibody complex. Once they have this complex, they will start to deposit it into the cells, okay, the target cells. And after that, they will mediate an inflammation or autoimmune responses. They're also utilizing the complement proteins. The neutrophil also involved in these reactions, and they can produce lots of granules or lots of mediators. More or less, it's quite similar to the type 1. Okay, it's just that they are forming their own antigen antibody complex. So these are the roots or the pathway or the schematic representation of the pathogenesis of immune complex mediated tissue injury, and the morphology consequences are depicted as the box area. Okay, so you start with the antigen binding to the antibody, and they start to form a complex among themselves, right, before they bind to any target cells. Part of them, okay, they also plus another protein, which is the complement protein that we have mentioned earlier, and also they are activating this protein for various activities like the neutrophil aggregations, which later on can cause the release of lysozymes enzyme, leading to the death of the cells. Apart from that, okay, on the other hand, it can cause anaphylatoxin generations, and this can manipulate or can cause a reaction to the blood vessels and changing or rendering the vascular permeability, leading to clinical consequences like the edema or vasodilations. It's all happening, I mean, based on the antigen, antibody, and also complement molecules. Antigen and antibody complex on its own can also trigger the platelet aggregation, leading to thrombus formation, then ischemic. It also can cause the activation of the Hashman factor. I think Hashman factor is the clotting factor number 6 or 12, I think 12. And later it will activate the kinin system, leading to edema and also vasodilation. So this is all the interrelation or the consequences of the antigen and antibody complexes. And we specifically put them in a separate section, which is the type 3 hypersensitivity. Immune complex vasculitis, the necrotic vessel wall is replaced by smudgy pink fibrinoid. Okay, this is an example at the cellular level. This is the histology. We move on to type 4, which is the cell mediated. It is mediated by can be single cell, right? And it's commonly associated or categorized under the delayed type hypersensitivity. And tissue reactions include the parenchymal destruction associated with perivascular lymphocytic and macrophage reactions. For the chronic, yes, this is true for the long-term consequences. Diseases like the chronic active hepatitis, viral exanthum. Viral exanthum is the eruptive skin rash that is often related to viral infection. And immunization or vaccination have the potential to decrease the number of cases of measles, mumps, rubella, and chickenpox. But all viral skin infections require clinical care by a physician or other healthcare professions. Contact dermatitis, inflammations of the skin cells due to the contact with some antigens present in some materials or some insects. Graft rejections is also type 4 or cell mediated because it occurs when the recipient's immune system attack the donor graft. Okay, when you are receiving and foreign organs from people or graft due to the transplantation, your immune cells start to recognize this and saying that, okay, this is foreign. Let us decide whether we want to destroy them or just accept them as part of the recipient's body. And the immune response is usually triggered by presence of the donor's own unique set of the HLA proteins, which the recipient's immune system will identify as foreign. All right. Other than that is the IBD, inflammatory bowel disease. It's also type 4 hypersensitivity. And this delayed hypersensitivity in the skin, as an example, reveals the perivascular cellular infiltrate that marks positively with anti-CD4 antibodies. You can see that lots of cell infiltrates, okay, cell that is invading the blood vessels, okay, after they've been stained with the enzyme and also antibody reaction. This is just histology for you to know at the cellular level what is really happening. This is a section of the lymph nodes showing several granulomas present, each made up of an aggregate of epithelial cells and surrounded by the lymphocytes. The granuloma in the center shows several multinucleate giant cells. You can see this is the granuloma, and also on the outer surface, you can see lots of epithelial cells, okay, here, and lots of lymphocyte here start to go and infiltrate into this granuloma. Okay. So it means that your body have a special mechanism. Always, they have different way, a unique way for them to cater or tackle this invading pathogen. All right, and type 4 and each of these types, they have different mechanism. If I ask you in the examination, explain type 1 hypersensitivity plus. Give me the examples of the diseases. So you can come up with the mechanism, you can come up with the images, you can illustrate the mechanism. How does it happen? This is schematic illustration of the event that gave rise to the formation of granuloma in type 4 hypersensitivity reactions. Note the role played by the T cell derived cytokines. Okay, you can see a bunch of a mixture of cells, a mixture of immune cells like lymphocyte, macrophage, normal cells like fibroblasts, epithelial cells, giant cells, natural killer cells, they all come to action. But each of these cells, they have their own way in tackling the antigens. It depends what type of signals that they receive. If the signals in the form of cytokines, so maybe macrophage is the best key player to regulate this. Okay, if they have the antigens, so the body will decide, okay, which pathway they are going, okay, which pathway that they are following. But the ultimate aim is to eliminate all these invading antigens. I didn't give you the type 5, which is the stimulatory, and I can say that it can be combined into any of these type, of hypersensitivity, and that's is very rare case. And I hope that all these type 1 to type 4 is enough for you to understand at your current level, okay? At least for this subject or for this topic. Let's move on to the second part of the lecture, which is the vaccines. I know this is a bit controversial, but we need to know what are the vaccine, the history, and what are the different types. What are the vaccines? The vaccine can be defined as the preparation, whether oral, injection, what are the route of administration, intradermally, whatever it is, okay, as long as we call it as a vaccines, they are intended to prevent the disease from keep on continuing or keep on occurring. So a biological is also can be defined as the biological preps that can be used to establish or improve your current health status, or maybe you want to get rid of certain invading or current disease that you are experiencing. It also can be defined as a substance that stimulates the body's immune response in order to prevent or control an infection. And the term vaccine derived from Edward Jenner in 1796 use of the term cowpox, in Latin term from vacca cow, which when administered to human provided them protection against smallpox. And vaccination is a cost-effective weapon for disease prevention. Okay, this is important for the disease prevention, but it's not the main aim. Okay, it's not the main or the only solution to treat or to prevent the disease from happening. There are lots of other alternative treatments and options that is possible to prevent the antigen or pathogen from invading your immune system. All right. So history, so that I can have a look at this, and maybe you can read through this a bit of history of the vaccine. And what is the vaccine really? Some okay, I can say some book they call it as a non-pathogenic antigen that mimics a particular pathogen in order to elicit an immune response as if as actual pathogens were in the body. In the case of COVID-19 virus, for example, the SARS-CoV-2, so you are taking this attenuated or the weakened virus SARS-CoV-2, and then you introduce into your immune system, and you want to see what would be the immune reaction. If you have a very strong immune system, of course, you can start to produce the antibody for the long-term recognition. Unfortunately, some people, fail to recognize this and also produce antibody, and it can cause a deleterious effect. So I cannot say that the vaccine is 100% safe for you as for now, because lots of clinical trials, lots of lab tests need to be performed before we can consider this as the absolute treatment for the viral infection or pathogenesis of any virus. And the overall goal of vaccines is to establish immunity against that particular pathogen. As I said, yes, you can use the vaccine to introduce the antigens so that your body become familiar with it. However, that's not the main solution. Okay. Types. We have two types of vaccine. We have the passive and also active immunization. Okay, if you considering the active one, you can see like the COVID-19, we have the weakened type. Okay, virus SARS-CoV-2, we have the killed one. They're all similar like the real life virus, but it's just that we make them weak so that they still preserve the antibody. So that's the the key point. The second generation vaccines, we have lots of different type. We have the capsular, which is more straightforward, more easy to produce. Maybe they have only taken not the whole virus, but maybe portion of the virus or certain protein. Toxoid. I think this is not toxoid. I think this is a typo error here. Let me try to fix it. If I can fix it, maybe not. It's okay. Okay. Toxoid. Okay, not. And we have the recombinant antigen vaccine. This is using the recombinant DNA technology. We manipulate some of the portion from the virus and only include that particular portion for your immune cells to recognize it and attack it, okay, and start producing the antibody. The third generation vaccines, of course, this is the upscaling or the newer version. We have the mRNA vaccines. We have like the one from the Pfizer. Some of the book says that this is not even a vaccine. It's a mRNA technology or gene therapy technology, or whatever it is all considered under the vaccine group. Okay, we have the DNA vaccine too. So different company are trying to study and try to make their own version of the vaccines and attacking or targeting a particular sequence on the virus, so that whenever the virus enters your body, they already have the memory and they already have the antibody to kill that virus. Active immunization means that introduction of foreign molecules into the body that causes the body to generate immunity against the target. And this immunity comes from the T cells and the B cells with their antibodies. This is the active immunization. Means the vaccine are used for active immunization too. Passive, we use the pre-synthesized elements in the immune system, and they can be transferred to a person so that the body does not need to produce these elements itself. Okay, we train the body to produce the antibody. And this method of immunization begins to work very quickly, but it's short-lasting because the antibody are naturally broken down. And if there are no B cells to produce more antibodies, they will disappear. So there's, they need help from our own body system. Passive immunization is done or is performed by injecting antibodies from an immunized animal or a human to a human patient. And it's very useful in case of urgent need to stop the toxic effect of certain disease like the snake venom bite, spider venom bite, botulism, okay, the tetanus, botox, okay, they're all the toxin. So it's important to stop or prevent the immune responses due to these toxin exposure. So you can see the example here from the venom of the snake. We extract it and low dilutions are injected into the horse. After 1 or 2 weeks, you can see that the horse are producing antibody against the venom from the snake bite. And then we extract this antibody and then we give to a snake bite victim in the human. Okay, because this antibody is produced in the horse. We use the horse as the host to produce the antibody. We don't, test it on human. What happens if the human cannot produce antibody? They will die. Okay, we are not allowed to do this in human, and we use the animal as a model, as a host to study human diseases. How cruel is that? I'm just joking. We don't have any other option, all right? So some of the examples of the passive immunization, you can see we already commercialized part of this, and in fact, we are using this antibody for the human use. We use rabbits, we use pig, we use what else? We use rats, we use what else? Here, yeah, horse, snake, as to study and to make the vaccine or to make the antibody. The causative organism or agent is grown in a very different environment from the normal environment. This causes organism to be weakened enough for immune response to attack and eliminate before it establishes disease in the host. For example, here Mycobacterium bovis. You grow in a high concentration of bile juice containing media for 13 years, and then you can produce the BCG strain that was created, which is used as a vaccine against tuberculosis. Okay, so people study the animals and also, testing this on different types of tissues and cells, and after that we can produce the antibody. So I think some of you, even me, receiving the BCG. So whenever we have or we got the Mycobacterium bovis invading our system, or Mycobacterium tuberculosis that causes the TB invading our system, we are already protected because we have this antibody against that specific pathogens. For the killed, inactivated, attenuated, weakened, so they're all the same thing, vaccines. So like the polio, so we inactivated them and produced the salt vaccine. Okay, the vaccine for the polio, this been used, more than 50 years, I think. And now I think in this year, I think early last year, we already we are being what we call being told that the new polio virus are invading the community in Philippines. And of course, when the new virus that emerged due to their mutations, maybe we need a new vaccine for that particular virus strain. Okay, because you know that the virus are evolving. Okay, they are mutating over the time, and of course, the vaccine that you provide or you produce today is not relevant. It's not suitable for the newly emerged viruses. Okay, there need to be a constant improvement on the vaccine production. However, not all people are suitable to take the vaccine. I keep on stressing that because due to the recent cases, lots of side effects that we can gain from the vaccination. Of course, we need more research and more data to conclude on this. The effectiveness of the vaccine in order to curb the viral transmission. If you compare the attenuated and also inactivated vaccine, they are different, but make sure yourself is not confused with this terminology. For the attenuated vaccine, selection for a virulent organism means that the virus is not the virus is no longer can causing any diseases. Okay, the virulent pathogen is grown under adverse culture conditions and the prolonged passage of virulent human pathogens through different hosts, and they require only single booster. However, they are less stable and they are humoral or cell mediated, and they may back to the virulent form, means they are not stable. So if the vaccine that you are taking are from the attenuated one, be careful because they can become active again, okay, in your system. Unlike these attenuated or activated ones, you can have the inactivated vaccine where they have this viral pathogens inactivated by chemicals or irradiation with gamma rays. And of course, you need them in a stages because you need the boosters keep on taking this I mean, maybe, quite a longer period of time, because you need them to become, spike, or you need them to become activated when you need them. And they are more stable and mainly humoral, not cell mediated, and cannot revert to virulent form. Then this is a good news, all right? And lots of people prefer this type. Another type is the capsular polysaccharide vaccine. They are using hydrophilic substances or polysaccharide capsules as one of their coatings. Okay, it targeting or making the antibody more stable. Okay, so that the macrophage and neutrophils easier to phagocytose or to kill them. So it's kind of helping these immune cells to kill them, right? The purified capsular polysaccharide vaccine, they depend on the status of your immune system. If your immune system have a robust activity of polymorphonuclear cells or phagocytes like macrophages, yes, you can have this one. But if you are immunodeficient, if you are immunocompromised, you are not suggested to take this type of vaccine. Okay, toxoid vaccines. Some bacterial pathogens, including those that cause diphtheria and tetanus, produce exotoxins, which produce many of the disease systems that result from infection. Diphtheria and tetanus vaccine can be made by purifying the bacteria exotoxin and then inactivating the toxin with formaldehyde to form a toxoid. All right, so this is the recombinant vaccine, how we learn from the virus structure, from the genome of the virus. We take only certain portion of the DNA and start to grow them in the yeast, and then we take the antibody from the yeast and start to give to the human. So there are lots of type of vaccines. I think you can look at here in the examination. Maybe I can ask what are these category of the vaccine and how it is their, the way that they make this. All right. For this one, maybe it's quite difficult for you to understand, but just for you to know how we can grow this recombinant vector vaccine, we're using the recombinant DNA technology to produce or to focus only on the certain portion of the DNA, not the whole viruses. The mRNA vaccine, the DNA vaccine, they are all being used, produced utilizing different ways, okay, different nucleic acid. Of course, you will need the double-stranded DNA to target a certain template DNA on the virus. If it's DNA virus, if it's mRNA, if it's RNA virus, maybe you want to consider producing the mRNA vaccines. It all depends on the viruses. You can look at my previous lecture. What are the DNA viruses? What are the RNA viruses? And that makes the vaccine production easier. So if you know the genome or the sequence of the DNA or RNA of the virus, then it's easier for us to make the vaccine. Okay. So this is just the similar things that I have already discussed quite early in the process. Okay, already discussed about this. Okay, so the mechanism of the vaccine, same thing. When your body recognize a different, they recognize the vaccine as the foreign antigen. And of course, we have these antigen presenting cells in our body as the front or the front liner in our protection, in our immune system. They are the first cells to recognize all types of antigens. And after that, they will pass on the information to the dendritic cells And after that, they will activate the T cells or helper cells, T helper cells or B cells to produce the antibody. All right. So I can, you already have this one, I think immunology one, you can always refer back to it. I don't want to spend too much time on these, innate and also the mediate, what do you call, innate and also innate and also adaptive immune response. You can have a look on this. Potential shortcomings of vaccines, as I mentioned, not all vaccines are effective because they are very specific, and of course they are different people with different immune status. So make sure you consult your physician before you take the vaccine. Make sure you are in a good health before you take any of the vaccines, not only for the COVID-19, but all other viruses. Okay, so this is just example how you can find the antibody spike once you have this antigen recognition. The first injection of antigen A, you can give a primary response. You can take like maybe 2 weeks, and after that it can goes down, and you start to give a booster, a second dose. You can see that it can increase again. So it's always depend. Okay, different type of vaccine can have a different mechanism, but this is only the schematic illustration to show that it depends on how well your body in producing the antibody. If your body capable to produce the antibody for a long term, yes, you don't need any booster anymore. But some vaccine is being produced in a way that you need to take the booster to complete the activity. Adjuvants is an ingredient that we use, okay, or we add to the vaccine to help them to stay longer, okay, stable. All right. In other words, the adjuvants help the vaccines works better. Okay, some vaccines we put it. All right. So these are the different routes of administration: IM, oral, intradermal, whatever it is. Okay, so it's for different purpose, and also you can elicit the effect for different time frame. Okay, this one is the booster. For most vaccines, the immunity against a particular pathogen has a tendency to wear off over time because they are not being triggered or not active anymore. So you need to give a second shot so that keep on spiking the antibody. So already discussed about this. This is also the summary, and also I give you some example for you to look at. What are the whole example of the whole vaccines? Okay, I put it Sabin polio vaccine for you to see the pros and cons. Okay, I keep on repeating this different type of vaccine just to recapitulate what I have saying earlier. All right, so this is again repetition what I've been saying before. So this is how we produce the vaccine in the lab. For you, it's not necessary, but it's just for you to know that this is the way that we grow the vaccine. Some vaccine being produced in the embryonated chicken eggs, and then also in the transgenic plant. We use the animal or plants as our host. For the immunological test, yes, you can know about this. We want to apply this for, especially for the diagnostic purposes, for you to know if a person having any like autoimmune diseases, or if they have, you know, any immunological reaction for a certain, you know, certain drugs or certain treatment. So we use this agglutination reaction. This is the most simplest reaction, the most simplest one, where we detect antigen or antibody in the patient specimen. For example, in the blood typing, the serology lab, okay, we're testing for antibodies to the infectious agent, testing for Haemophilus influenza type B capsular antigen in the cerebrospinal fluid. We use this antigen and antibody reaction as part of this activity at patient serum, and then see if the particles clumps. Okay, this is an example. You already done this. Maybe in the serology lab, you know that the patient, they're adding the blood for blood grouping, for the reserve. So you look at the clumps. Negative, no clumps. Okay. Positive, you can see the reserve is positive. That's agglutination. So they're utilizing the antigen and antibody concept to say or to group your blood. So in this case, you see that the A, B, and results, you can see agglutination occur in the A, and results also positive in the results plate. We have another test that we can use in the lab. We call the direct Coombs test. We perform this in patient with a hemolytic anemia. We want to detect the antibody or the complement system on the patient's red cells. I'm not sure what is the C, but detection, I think it's the complement one. Detection of antibody. Okay, the presence of the antibody. What type of antibody? Is it IgG, IgE, IgM? Okay, we use the patient's RBC, we coat them with the antibody, and then we add the anti-human globulin or Coombs reagent. Okay, this is another name, and we look at the agglutination. We take the RBC plus this anti-human globulin, and then if there's any agglutination, then we can say that the patient red cells are coated with the antibody or complement. Yes, complement. The C is the complement. So this is the Coombs test, one of the diagnosis. IAT is indirect Coombs test. We perform as part of the pre-transfusion testing for the cross-matching or antibody screening before you do the transfusion, maybe before you do any transplantation. Maybe this is one of the things that we look or we do or we perform in the lab. We use the patient serum that have the antibody. We add the donor red blood cell that is coated with the antigen. After that, we add the AHG again and look for the agglutination. So positive test result means agglutination occur. Okay, antibody screening, you can see if there's any agglutination, again, that's positive. Clumping means that the patient has the antibody to the donor or to the donor red cells. I mean, they have this type of antibody. Immunofluorescence, usually we use in the lab to detect bacterial presence or any formation of antibody antigen complex. We use the IF or immunofluorescence method. We use a direct and indirect. I think this is not important for you because this is used in the cell culture lab where we want to differentiate between two different cells. We use the immunofluorescence detector to interpret or to check whether the cells producing a certain antigen that you are studying. ELISA, or in other words, I think enzyme link immunosorbent assay. Also, we use in the lab, in the serology lab, to check for any presence of a protein for certain diseases. They use the absorbance method to check for the color differences change. So if the patient have the antibody or the desired antibody, you can see that the color is changing. Sandwich immunoassay. So this is only the variations of ELISA method. You don't need to know this in detail, but just to show you that this is the things that we are using or we are applying in the pathology lab and also serology lab in the hospital, basically. So the Western blot is another protein detection, antibody detection. Most common, we use the HIV test to confirm the presence of this human immunodeficiency virus. Again, we use the agglutination. Not agglutination, sorry, we add the anti-human globulin, but we look for the expression or the bands. Okay, the expression of the band, the formation of the protein, how big is the protein being produced? Okay, does the protein being produced the antibodies against the HIV or not. If you see the presence of this IgE, for example, against the HIV, then you can see that the patient are HIV positive. So this is the band that we are talking about. Enough band positive is patient is positive, right? A, B, C, you can see that the patient in C is, expressing all the HIV protein, okay, all the viral protein that is present in the HIV virus. 1 and 2 is only the control, okay? Flow cytometry also we use in the lab, but it's not for you because it's only for the research lab. You just send the sample, okay, the blood sample from the patient or biopsy to the lab, and then they will do all these tests. The flow cytometry, basically, they use the cytometer to measure the size of the cells, and also looking at the protein that is expressed on the outer layer of the cells or the surface marker proteins. If the surface marker proteins expressed in that particular cell have this antibody binding to it, so you can see, okay, you can conclude that this patient have a cancer. You can conclude that the patient has the autoimmune diseases and so on. Okay, so I think that can conclude the today's session. Apologies because this is taking a lot of your time, but you can have the, you can have the slides or lecture I will provide to the next after this. If you don't understand, because I go quicker towards the end of the slides, because I think this is some of them is not important at your stage. Okay. However, if you got any questions, any ambiguities, if you are not clear from the slides, please let me know so that we can have any replacement or revision class after this. See you again. Make sure you watch these recordings today, and also look for the and sign in record your attendance to using the link that I provided to the NESA. I think that's it. Let us end the session by tasbih kafarah and surah to us. Subhanakallahumma bihamdika asyhadu an la ilaha illa anta astaghfiruka wa atubu ilaik. والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. Till we meet again, this video will be available in my YouTube channel. Thank you very much, everyone. Assalamualaikum warahmatullahi wabarakatuh. Bye bye. See you later. Bye bye.